So our study concentrates on the TP53 mutation landscape in chronic lymphocytic leukemia and its prognostic impact. It's a multicentric retrospective analysis that included more than 10,000 CLL patients from our single institution and from 39 prospective clinical trials in order really to investigate how TP53 and all its intrinsic variables can affect prognostic impact in CLL, also in respect to other genetic markers and the kind of therapy that patients received. So it's really a translational research project that aimed to bring all the genetic results that we could get at our single institution academic facility in Ulm from the bench to the bedside. So we selected these 10,051 CLL patients. Two-thirds of them were sequenced for TP53 via Sanger sequencing and DHPLC, and one-third via next-generation sequencing. Afterwards, we really planned to, and we did, perform the pathogenicity prediction and a variant interpretation following the current guidelines of the European Research Initiative for CLL that have been published on leukemia in 2024, and also based on the UMD-based tool CECHAT, an informatic tool for correct TP53 assessment. And thanks to these informatic tools and to the guidelines, we could actually interpret TP53 with regard of its pathogenicity, the TP53 activity, its location inside and outside the DNA binding domain, the gain and loss of function, and also the variant of allele fraction. Yeah, so first of all, we could confirm in our efficacy cohort of previously untreated patients, 3,713 patients that we selected to correlate the genetic findings to the clinical result. We could confirm, so in this huge population, that TP53 mutations are prognostic for PFS and OS, and that they are standalone prognosticators in CLL. In fact, we also looked at mutated TP53 in absence of a 17p deletion, and we could confirm that even as independent prognostic factor, it had a significant impact on PFS and OS. And then we also looked at TP53 in unmutated and mutated IGHV status. And in both cases, it could retain its prognostic significance. What's also very interesting and curious about our analysis is that we really looked at TP53 intrinsic and specific parameters and correlated them with outcome. What we found is, for example, that variants leading to a partially active TP53, they do not really have an influence on PFS and OS and show a survival curve that is similar to wild type. This is a finding that has been scarcely, if at all, described in CLL patients. We have some data regarding mouse models for solid oncology, but not yet in CLL patients. So this was surprising me, for example, as a result. And also we looked at splicing mutations in particular, and they also had the worst outcome also in comparison to missense mutations. And this would be also a finding that was scarcely described in CLL literature. So our next step would be to really look longitudinally on how TP53 mutated cohorts behave under therapy. So we really had a look into into 3,700 and more previously untreated CLL patients. Now we want to really have a screenshot, a snapshot on how they behave in a relapsed refractory setting. If TP53 sees a dynamic and how this dynamic is.